Hello everyone. Today we are going to discuss the syllabus of Microbiology Part 1 for the first year students. Before moving towards the syllabus, it's very important to understand what is microbiology. If we break the words, micro means small or minute, bio means living, and logy means study. Microbiology in simple terms is nothing but the study of smallest living organism with the help of microscope. For first year, since it's a credit system, each subject consists of total 5.5 credit points, in which if we consider or if we talk about microbiology subject, part 1 consists of 2 credit points, which is introduction to microbial world. Part 2 also consists of 2 credit points, which is basic techniques in microbiology, and 1.5 credit points are given to the practicals, which are completely based on the theory paper 1 and 2. When we talk about the theory paper 1, which is introduction to the microbial world, the name itself indicates it gives us the brief introduction about the microorganism and the study of microbiology, its history, inventions, different theories, and lot more. We will discuss all the points mentioned in the syllabus one by one. There are two credits, or in simple language, two chapters, in which first important topic is Amazing World of Microbiology. The first important part or topic in this chapter is development of microbiology as a discipline, which actually tells us how did the microorganisms were found for the first time. This for the study of microorganism was the very important equipment required was microscope, and this microscope was discovered by Anton von Leeuwenhoek and Robert Hooke in near about 17th century for the first time. Anton von Leeuwenhoek is also known as father of microbiology, who for the first time just gathered a lenses together and observed a small living particle which was a living organism. Since then, the microorganisms were not introduced, so he named them as an animalcule. And further, Robert Hooke, after the discovery of microscope, started the study of the microorganism. All his theories we will discuss in detail in our further videos. Then, further, the theory of abiogenesis and biogenesis. The theory of abiogenesis is also known as spontaneous generation theory, which was proposed by Aristotle. Scientist Aristotle proposed the theory of abiogenesis, and he stated in that the living organism is produced from the pre-existing non-living organism. And for this theory, he proposed different experiment. But scientists Louis Pasteur, Francisco Redi, and Tyndall, they performed various experiments to disprove this theory, and after due to getting different proofs of their experiments, they proposed the theory of biogenesis, in which they stated that the living thing which is produced are generated from the pre-existing living organism. For this, Louis Pasteur performed an experiment with sonic jar. Then Tyndall performed the experiment of Tyndallization. These experiments also we will study as the points are moved forward. Then the next important part in our syllabus is golden era of microbiology. Why the name is given golden era? Because in this time or in this period, the vast improvement or development were taken in the field of microbiology. Various scientists contributed for the development of microorganism, their study, their characterization. were done in this period. So, this period is known as the golden era of microbiology. In this, the first scientist who contributed for the study of microorganism is Louis Pasteur. Louis Pasteur performed various experiments. He has introduced the technique of fermentation. What is fermentation? Fermentation is nothing but conversion of sugars into acids and alcohol. When we consider regarding microbiology, Louis Pasteur used different types of microorganisms, especially yeast, in the conversion of alcohols or in beverage production for the fermentation process. Then he also introduced new technique of pasteurization. Pasteurization he used in fermentation process for increasing the shelf life of beer, and now this technique is also used to increase the shelf life of milk. He introduced different types of vaccines for rabies and cholera. During this study, Robert Koch he studied on the various diseases caused due to microorganisms and proposed the four important postulates depending on the characteristics of the diseases. He stated that in every case of infection, the microorganisms are present. These microorganisms can be re-isolated from the diseased person if they are inoculated in the uninfected host. They can cause the same disease again, and for proving this, he stated the four important postulates in the case of bacterial diseases. In the same way, reverse postulates were proposed for the diseases or infections caused by the viruses. Robert Koch also performed the staining technique. Now, what is staining? Generally, the microorganisms are transparent, and as we cannot see them with the help of naked eyes, in microscope also there are less chances of their visibility. So it is very important to give them color. Staining is nothing but we generally give them color or dyeing technique in simple language to make the microorganisms visible. To study their morphological characteristics, the staining is used. What is staining and the types of staining we are going to study in detail in part second and also during your practical courses. Then the next important part is the discovery of viruses. As the viruses are difficult or impossible to grow in lab media because it requires a particular cell, the discovery of viruses were the very important discovery for knowing the diseases caused by viruses. For the first time, scientists Ivanovsky studied the disease caused by viruses on the tobacco plant, and he named it as tobacco mosaic viruses. For the presence of viruses, its route of infection, he performed the various experiments, and then he came to know a slimy structure, the main virus. He named it as virus because in Greek language, virus means a slim thread like structure, which he observed in the solution which he have taken from the scraping of the leaves. Next contribution was of Joseph Lister in the antiseptic surgery. As we know, during the surgery, different types of tools are like scissors, knives are used for the surgery purposes, and if the same tools without sterilization are used for the another patient, there may be the chances of infection to the patient. So, to avoid the contamination or the harmful effect caused by the organism present in the previous patient, there was a need of sterilization of specific tools used during the surgery. And for the first time, Joseph Lister had sterilized all these tools, and these tools were used to perform the antiseptic surgeries. Then the Paul Ehrlich performed the theory of chemotherapy, which is used in the treatment of cancer. Further, E.L. Metinchuk proposed the theory of phagocytosis. Here, phagos means engulf and cytosis means cell. The process of engulfment of whole cell was performed by particular microorganism, and these whole process were studied and proposed by E.L. Metchnikoff. Further, in the field of medical science, microbiology and immunology, the development of vaccination by Edward Jenner and penicillin by Alexander Fleming had brought a great success for treatment of diseases caused by microorganisms. The discovery of penicillin was an accidental case of by the Alexander Fleming when he left his experimental remainings back and due to his failure in his experiment. After few days when he observed the plates, he found the zone of inhibition around some colonies. Now what is zone of inhibition? It is a clear zone or circle observed in around the colony where another organism was unable to grow. He performed similar experiment for number of times. Now how he performed it and what were the results, that we will study in detail as the points will go forward in the another videos. But the production of penicillin and vaccinations were very important aspect in the medical microbiology. Then the growth of microorganisms in lab is performed by using different enrichment media or cultural media which are high which consist of high content of nutrients which will enhance the growth of microorganism, and these different enrichment culture techniques were developed by scientist Martinus Belgenic. He contributed in the development of different cultural techniques. He developed different types of media specific for the specific organism and also for the rhizobium. Then in the field of soil microbiology, the scientist Vinogradskaya had developed a Vinograd sky column to check the actual activity of different microorganisms for the nitrogen fixation and chemolithotropy. And during this development, the golden era of microbiology came to an end, and further, the modern era of microbiology came into existence. Why it is known as modern era of microbiology? Because in this era we actually used the different technologies for the study of microorganisms, their activities and their characteristics. The scientist Carl Woese, based on 16S RNAs, studied the applications and significance of different microorganisms such as human microbiome. Now, what is human microbiome? The microorganism present on or within the human tissue or body fluids, for example, a skin, gastrointestinal tract, mucosal membrane, etc. These microorganisms together, or aggregate of these microorganisms, is known as human microbiome. Then the next term was nanobiotechnology, or it is also known as bio-nanotechnology. It is nothing but intersection of nanotechnology and biology, and it is often used to describe the overlapping of multidisciplinary activities associated with the biosensors or biology or biophysics or biomedicines, etc. Simply using the nanotechnology in the biological agent to produce any particular tool, such as for example biosensor. And the next very important part is space microbiology. Now, space microbiology is also known as astromicrobiology or exomicrobiology, and it is nothing but the study of microorganisms in the outer space. Now, the space microbiology or astrobiology efforts are aimed at understanding the origin of life and the search for the life other than Earth. The search for extracellular microbial life have focused mostly on Mars, but other sites also includes the different types of moons like Europa, Titan, or Enceladus. These are nothing but the names of different moons. On these moons also, now the scientists are trying to find the existence of any particular living organism. It may be microorganism or animal or any plant, and these studies are carried out in the space, so it is known as space microbiology. Here, all these points we have to study in the modern era of microbiology. Then the next point is Nobel laureates in the life science of 21st century. All the Nobel Prize winners in the field of life science, it may consist of microbiology, botany, zoology, any field which is related to life science. In the 21st century, the scientists, those who have got Nobel Prize, we have to make a search and a report on their research. These all points are included in Credit 1, which is Amazing World of Microbiology, and all these points we will discuss one by one in our different videos. Second credit is Types of Microorganism and their differentiating characteristics. Now its name itself indicate, in this chapter we are going to study about the different types of microorganism and what type of characteristics they are going to consider. The first point is prokaryotes, eukaryotes, 3 domain and 5 domain system of classification. In 11th and 12th you have already studied the 5 kingdom system, then the different 3 domain system, but here we are going to study the same with the help of their characters. Also we are going to study the different types of organisms like bacteria, protozoa, fungi, algae, viruses and actinomycetes. When we consider about their differentiating characteristics, which may include their morphological characteristics, their anatomical characteristics, their genetical makeup, habitats, and so on. These all the things we are going to study in detail. Now there are terms viroids and primes. Now what are viroids? Viroids are the mature viruses which infect the plant cell, or the disease causing viruses of plants are called as viroids. Then what are prions? Prions are not actually viruses, but they are abnormal proteins which are produced during the protein synthesis because of any particular mutation or abnormalities. These prions directly affect the neurological system. They break the connection between the neurons, which leads to the imbalance of particular animal. For example, it may cause mad cow disease in which the cow loses its neurological signals and it started behaving weird, like tapping of feet or jumping, and other different characteristics are seen. Next important part in the credit second is beneficial and harmful effects of microorganism. Now when we pronounce the word microorganism, generally the people are knows only the harmful effects caused by the microorganisms, but there are a number of microorganisms which may help us to perform various activities. The different types of microorganisms are used in various industries and even in our day-to-day life, which helps us to perform or in the formation of various products. In this, we are going to study first part is medical microbiology. When we see the medical microbiology, there are number of infections or diseases, or in every case of infection there is presence of microorganism, or one can say each disease is caused due to presence of microorganisms. So this is its harmful effect. But to cure these diseases, different vaccines or antibiotics are produced with the help of microorganism. Now what are antibiotic? The word antibiotic, anti means against, and biotics means living organism. The substance which is given against any particular living organism is called as antibiotic in simple language. Now how these antibiotics are produced and list of different diseases caused with their causative agents, means with microorganisms, we will see during the lectures. Next point is environmental microbiology. Now the environment in this case means soil, water, air, and also all the sediments covering our planet. It can include animals and plants that inhabitat in these areas. When we study the environmental microbiology, the first point in this is eutrophication. Now, what is eutrophication? Due to heavy rain or floods, the soil from different agricultural areas is directly carried out in ponds or lakes, and due to this, the ponds and lake water becomes enriched in minerals. When this pond water get enriched in minerals, there is heavy growth of algal blooms. When these blooms are grown heavily, they deplete the oxygen content of water, and due to this, the other water body animals like fishes are unable to survive, and this whole process is known as eutrophication, or it is also called as hypertrophication. When or it can be defined as when the water body becomes overly enriched with minerals and nutrients which induce excessive growth of algae is known as eutrophication. The next point is red tide. Now, what is red tide? Red tide is heavy growth of red algae in the saline areas. And these red algae are highly toxic in natures, and generally it is found in oceans and seas. The next important point is sewage treatment and bioremediation. Some different types of microorganisms can be used in the treatment of wastewater left from different industries or sewage domestic water, or which are left through the sewage lines. Then bioremediation. Bioremediation is nothing but the process used to remove the contaminants or pollutants included in water, soil, or subsurface area. In this, we alter the environmental condition which stimulates the growth of microorganisms. When there are heavy growth of microorganisms, this microorganism helps to degrade the pollutants present in the environment, and by this way we can remediate or treat the environment to remove the pollutants. Then the next topic is food and dairy microbiology. The name itself indicates the there are different of food types of food products which get spoiled with the help of microorganisms, and if we eat this spoiled food, the number of foodborne diseases are caused due to microorganisms. But they not only cause the spoilage of food, but for the production of different fermented food products, microorganisms are very beneficial. For example, the bakery products like breads, cakes, the yeast is mostly used for the production of these bakery products. So in food industries, the microorganisms are very beneficial. And when we consider about dairy microbiology, there are number of milk products like curd, cheese, buttermilk. These are prepared with the help of microorganisms at the faster rate. Next point is agricultural microbiology. Now, when we deal with agricultural microbiology, it is very important to increase the soil fertility to increase the crop yield. And for increasing the soil fertility, we use different types of fertilizers. But due to development of different types of technology, nowadays the microorganisms can be directly used as a fertilizers to enrich the soil quality, and this will automatically lead in the increase of crop yield. But different some type of microorganisms like bacteria or viruses are responsible for the different plant diseases also. And for these type of infectious agent, some microorganisms are used to control their growth, and such agents are known as biocontrol agent. Any particular living organism used to control the growth of another organism is known as biocontrol agent. And these all points we are going to study in detail later. Next is industrial microbiology. Now, when we deal with industrial microbiology, it completely deals with the production of different types of products. Now, when we produce product, the main aim of the producer is to yield maximum product. It may be for the production by considering vaccines, it may be beverage industries, it may be food and dairy industries, it may be pharmaceutical industries for the production of antibiotics or different types of vaccines, and different types of enzymes are produced. Now, what are enzymes? Enzymes are nothing but biocatalyst which enhance or increases the rate of reaction. And during the product reaction, it is very important to convert substrates into product, and these enzymes help the rapid conversion of substrates into product by without altering itself or without undergoing any changes. Then different types of solvents are also prepared with the help of microorganism, but there is a vast chances of contamination or addition of pollutants due to the presence of microorganism which may affect the quality of product. Next, and the last important point is immunology. Now, immunology is completely based on the immunity. Immunity is nothing but the resistance developed by the host to any particular infection. In this, we are going to study the normal flora of microorganism in different infections or in different areas, and we are also going to see the three line of defense. Now, three line of defense are first line of defense, second line of defense, and third line of defense. The name itself indicate defense means we are helps us to stop or to prevent our body from the different infections. Now, first line of infection also consists of different types like physical barrier, physiological barrier, anatomical barrier. For example, skin, mucosal membrane, coughing or sneezing. These are nothing but included in first line of defense. It is just like, for example, a military force which are standing on the border, which prevents a foreign particle to enter in our body. In similar way, the second line of defense acts after the entry of microorganism or infectious agent or foreign particle inside our body. Then third line of defense, different types of cells or cell mediated immunity acts against the microorganism by producing different types of antibodies against the antigen. And this everything, all the three line of defense, we will study in the detail in the further lectures. These all points are included in your microbiology part 1 syllabus. Thank you so much.